Well, thank you very much for the invitation to present the work and also for the great introduction to the xCELLigence technology, which I'll be telling you about today, how we're using that also with our stem cell stuff and with regard to our drug development efforts to develop drugs to reduce metastasis. So I'll be talking about how we've used xCELLigence technology for all three of those approaches. To give you more background on the project itself, we're specifically studying the reattachment of circulating breast tumor cells. And so to think broadly about the problem of breast cancer, for breast cancer, like many other patients, do not typically succumb to the primary tumor, which can be removed by surgery, but are really threatened once tumors recur metastatically. And the great challenge that we currently face in the clinic is some of our best imaging methods for detecting tumors being MRI, PET-CT, or mammogram. Really, a tumor in a patient, once it reaches a foci of more than 10 million tumors. So we're really seeing cancer at very late stages. Now, it's been appreciated for many years that primary tumors will shed circulating tumor cells, or CTCs, into the bloodstream at a very early stage. And that because tumor cells are too large to fit through capillaries, they rapidly get stuck in the capillary beds of distant tissues. Some are pushed through and fragmented by blood pressure, but a subset of these cells can escape the blood vessel and persist dormantly in the distant tissue. But because these cells are not undergoing cell division because the environment is wrong, their dormant persistence, they're very difficult to think chemotherapies that target dividing cells. So these important stages of seeding and dormancy of metastatic tumor cells exist below our current clinical detection threshold, not effectively treated by our current chemotherapies that target tumor growth. Now, in the lab, we can follow these stages of early metastasis using tumor cells that express firefly luciferase, so that when these cells are injected into the bloodstream of mice, within minutes you can see that the cells have trapped within the capillaries of the lungs of those mice. Over the first few days, you tend to lose somewhere greater than 90% of the tumor cells as they're pushed through the capillaries. But then eventually, over the course of weeks to months, you get a reoccurrence of these tumor cells in the lung so that you can see the eventual metastatic outgrowth. So the point here being that in mice, we can follow the initial trapping, the dormant survival, and the reoccurrence of the circulating tumor cells. So many other labs are interested in learning ways to eradicate these cells at these early stages to delay or eliminate metastatic recurrence. Came into this project the dynamic responses of circulating tumor cells to the free-floating environment of the bloodstream. And an observation that we made when we took mammary epithelial cells and put them over a surface that they couldn't attach to, we noticed that the cells generated these long extensions of the cell surface that we hadn't really seen before. You're seeing the movie in this case in the presentation. Is that right? Anyway, I hope people are seeing that. We see these long extensions of the tumor cell stretching out. Now, my experience prior to this was looking at rearrangement of the actin cytoskeleton. And so if we look at how the actin site is mammary epithelial cells as they attach to extracellular matrix, so these are two mammary epithelial cells in red with phalloidin for polymerized actin, and in blue for DNA with DAPI. And so what you can see is the cells spread on extracellular matrix, they generate these broad actin membrane ruffles, or these spike-like filopodia. So we initially thought that maybe these projections we were seeing on the free-floating cells were actin-based filopodia. So we did a relatively simple experiment. We took the cells when they'd only been detached and free bits, and it formed these short extensions on their surface. We treated those cells with an inhibitor of actin polymerization known as trunculin A, which is known to destroy actin and betapodia. And we made the prediction that if these extensions were based on actin, that inhibiting actin polymerization would suppress them and cause them to collapse, because this was known for filopodium and betapodia. So we did that experiment, and we got a completely unexpected result. The projections did not go away. In fact, they got longer and much more numerous. That's that they were not based on actin, When we followed up with immunofluorescence here in the same cell via phase contrast, immunofluorescence for alpha-tubulin or actin, we could see that alpha-tubulin and microtubules were along the lengths of these projections, but actin was really only found at these dense ends and not so much along the length. So looking at a structure that hadn't been reported before, and we named them microtentacles for reasons that I'll explain in a second. One of the main reasons that we named them microtentacles was because these projections seem to play a role in how tumor cells aggregate together. So most human solid tumors are epithelial carcinomas, and epithelial cells in the body are tasked with barrier function. So the cells will make aggressive attempts to attach to each other. And so what you're looking at here is a confocal of two breast tumor cells free-floating and attaching to each other. We've labeled their plasma membrane with a red plasma membrane dye. And so we can see that the cells are forming tentacles. We can see that they're attaching to each other, but we can't really tell which cell is which. In case we labeled the cell to specifically track its projections. And what we could see there was that that cell is extending tentacles around its neighbor that are helping the two cells hold together. So we followed up these studies by looking at panels of metastatic breast tumor cell lines and ones that were more weakly metastatic. And we saw that the more cell lines tended to have more of these tentacles than the ones that were less aggressive. Around the same time, there was also a report from another group, Garth Nicholson's group, where basically they had looked at circulating colon carcinoma cells using intravital microscopy. So in this experiment, they used fluorescently labeled with calcine AM that are injected into the portal vein of animals and then imaged in the liver sinusoidal capillaries by directly putting a microscope objective onto the liver of the animal in this case. So what they can do here is they can look at the fluorescent tumor cells as they attach to the blood vessel wall, migrate into adjacent tissue. And so what they did was they followed the circulating tumor cells over 30 minutes in the circulation and counted the number of cells that were able to attach to the blood vessel. And what they saw in this study that was interesting was that if they inhibited microtubule polymerization with nab-paclitaxel, they prevented the ability of the circulating tumor cells to attach to the blood vessel wall. But even more interestingly, if they inhibited actin polymerization, which most people think for how tumor cells move around on a surface, if they inhibited actin polymerization, more tumor cells actually bound to the blood vessel wall. So this was in their study, and although they were looking at a very macroscopic level here, so not very much subcellular detail, as they're describing that microtubules are required for attachment, and actin depolymerization, actually attachment of circulating, match almost exactly with the mechanism that we were describing for microtentacles, dependent on microtubules and actually repressed by actin. So we followed up on this, again using microscopy of a GFP-labeled tumor cell attaching to M-cherry-labeled endothelial cells to model the reattachment of tumor cells to the blood vessel wall. And so with confocal microscopy here, we can see where the tumor cell is penetrating through the endothelial layer, but we can also cut through in cross-section and see that the tentacles are actually penetrating the junctions of the cells. All right, so if we bring this back to where we think tentacles are playing a role during metastasis, we know that primary tumors will shed millions of cells into them every day. The question of significance to patients is, what are the fates of these circulating cells? Well, thankfully, metastasis is a very inefficient process, and many of these cells mitosis when they're released into blood. Other cells will be fragmented by blood pressure or destroyed by the immune system. The cells that can escape the vessel do so by attaching via a mechanism that matches microtentacles, and then using actin-dependent crawling to pull the cell, but that you need both of these steps to successfully form a metastatic tumor to allow the cells to escape and grow out as a metastatic tumor. And where our lab has been fortunate is there are literally hundreds of labs looking at actin-dependent motility of tumor cells that are attached to surfaces, but they're looking at what free-floating tumor cells are doing. And here's where we can inhibit microtentacles. We make these cells from reattaching in the first place and force death by fragmentation. So that's really the underlying goal of my lab. And so in order to identify targets and investigate this more rigorously, over the last few years, we've been able to develop a sort of a current working model for microtentacle structure. And so basically there's a network of actin filaments that lies beneath the plasma membrane called the actin, which exerts a contractile force inward on cells. This is counteracted by microtubules growing from the microtubule organizer, which grow outward toward the cell surface. And these microtubules tend to contain a tyrosine of alpha-tubulin, which marks them as a fairly rapid and dynamic microtubules that persist for only about 3 to 5 minutes in cells. So they're constant, but it's this balance of microtubule extension outward and actin cortical contraction inward that gives the cell a stability. Now, recent work has shown that removal of this tyrosine through carboxypeptidase yields D-tyroglue microtubules, which now have a glutamic acid residue at the C-terminus of the alpha-tubulin. And rather than having short-lived 3 to 5 minute microtubules first, this was work that was done largely by Greg Gunn. Microtubules persist through intermediate filament protein via kinesin motor proteins. And it's the cord filament types via kinesins that we see that serve tentacles. So these two filament systems working together have the extension strength to overcome the contraction in the actin lead to the formation of a microtentacle. So while the carboxypeptidase has not been well characterized yet, the enzyme that catalyzes the reading to the glue microtubules, tubulin tyrosine ligase, has been well characterized. This will come up again when we talk about EMT, the tubulin tyrosine ligase will. All right, so it's always nice to see this as a cartoon model. Microscopist, I always prefer to see these processes dynamically in the cells to gain more confidence in what you're looking at. So what you're seeing here is a confocal microscope image of a mammary epithelial cell that's attached to it's been treated with a GFP fusion protein, EB1, that binds to the growing ends of microtubules. And so the movie that I'll show you here has about a 5-second frame rate. So microtubules are moving very rapidly. And what you can see is that microtubules generally proceed outward from the cell center. And where they encounter the edge of the tense, where actin is tense, microtubules get diverted along the edge of the cell. But where actin is rearranging or actin is looser, now and extend the cell surface. If you look really, you can see that there's one that's pushing outward, but it's gradually being pulled back by the actin contraction there. So in a cell that's attached to extracellular matrix, the actin cortex can tension the outer surface of the cell and large microtubule extension. But when the cell is detached from the extracellular matrix, it loses much of its ability to tension the actin cortex, and now microtubules can extend outward more freely. And this is really the underlying balance that we think is leading to the generation of microtentacles. When the cell is free-floating and detached, microtubule extension strained by actin cortical contraction. So understanding some of these mechanisms that are supporting microtentacles to identify several different therapeutic strategies. This is one here that was published by a graduate student in 2011 using tetracaine, which is a local anesthetic, which targets the kinesin motor proteins. So looking at a movie here of a cell forming tentacles over about 10 minutes, we can see the motion of the tetracaine takes effect, the tentacles collapse to the cell center. And I'll play that one more time. The reason that the tentacles are collapsing is because the tetracaine removes the ability of the vimentin and the tubulin to be coordinated by kinesins. So the vimentin collapses, and then the residual cortical contraction of the actin cytoskeleton pulls the microtentacles back in. So watching the movie again, you'll see that right about here, they begin to be pulled back in to the center of the cell. All right, given the role of vimentin, the other phenomenon that we were interested in looking at was epithelial to mesenchymal transition, where there is a strong upregulation of vimentin that occurs during this process. So we worked together with Jin Yang from Bob Weinberg's lab, using human mammary epithelial cells that either express a vector control or express the transcriptional activators TWIST or SNAIL, which are well known to induce an epithelial mesenchymal transition. which in the attached cells shows this transition from an epithelial morphology functions to the more spindle-like EMT morphology in attached cells. And again, all we did here was take these cells and free float them, and what we could see was that the cell EMT via twist or snail formed many more tentacles than the cells that expressed the vector alone. And that blinded quantity showed a 15 to 20-fold increase in microtentacle levels. When we paralleled this with protein immunoblotting, again for the vector, twist, and snail cells, you could see that the classic EMT transitions occurred. We lost E-cadherin, gained N-cadherin, gained vimentin, firm expression of twist and snail relative to a beta-actin loading control. So the regular EMT markers were changing. But when we looked specifically at microtubule regulation, we could see that the stabilized blue tubulin went up in the cells that expressed twist or snail, relative to total altitude tubulin was increasing. And this was specifically linked to downregulation of the tubulin tyrosine ligase enzyme, which recycles. So this identified a potential mechanism here for how you get increased microtubule stabilization during EMT, and regulation of the enzyme that destabilizes the microtubule. And so this was also one of the first times that we used xCELLigence technology to gauge the reattachment efficiency of these cells from the. And by comparing cells expressing GFP, snail, or twist, what we could see was that the twist cells reattached over this first hour much more efficiently than the GFP control cells, and the snail cells also more efficiently. So the strength here of the xCELLigence technology is it really gave us the ability to take very rapid measurements over time measurements that then allowed us to see these differences in reattachment efficiency that we could follow up with more complex plate-based assays, reattachment efficiency of the twist and snail cells, but then also identified for us time points that we could follow up with the live cell confocal microscopy to show that the tentacles were allowing the cells to reattach to endothelial layers at certain time points when the xCELLigence technology had told us sort of the early reattachment efficiency benefits for the EMT cells. We also took this back to patient samples where we looked at ductal carcinoma in situ, which is a very early stage of breast cancer. And we looked at expression of the EMT marker or the stabilized glue microtubules in parallel sections. And so what you're looking at here are the tissue sections, and the brown staining is the immunohistochemic twist or glue tubulin. And so with these DCIS tumors, well encapsulated, so they have this strong purple basement membrane that surrounds them. And that within the center of the tumor, expression of twist and glue is fairly low. But that if you look carefully at where the tumor is beginning to invade adjacent tissue here, and we look at higher magnification here in the second set of panels, cells that are pushing into adjacent tissue have begun to turn darker brown for both the twist EMT marker and the stabilized glue microtubules. And this is just another example here where the center of the tumor is fairly low staining for twist and glue, but where the tumor cells are invading adjacent tissue, very dark brown indicating EMT and stabilized microtubules. So this is telling us that as tumor cells begin to invade, they're undergoing the EMT changes and microtubule stabilization to help them with metastatic reattachment once they make it to the bloodstream. Okay, and so then very recently, just in the last couple of months, we published in Cancer Research that microtentacles are also increased in cancer stem cell populations. And we were really led to this by the connection between EMT stem cell phenotype that had been shown by Bob Weinberg's group and other groups. And so what we did in this series of experiments was we took the human mammary epithelial cells that had clearly distinct CD44, CD24 flow cytometry, distinct subpopulations of breast tumor stem cells that are 44 high, 24 low, and non-stem-like cells, which are 24 high, 44 low. And so Monica Charpentier in the lab, an MD PhD student, actually used flow sorting to separate out these and showed that microtentacles were significantly higher in the cancer stem cell population as compared to the non-stem population, and that these populations also formed more mammospheres when plated into a classic stem cell assay to form mammospheres. So the stem-like cells form more tentacles and mammospheres. Monica also looked at reattachment with the xCELLigence technology. And so the stem-like subpopulation was capable of reattaching much more quickly than the non-stem-like population. When she analyzed these individually, the stem-like cells attached rapidly, and the microtubule depolymerizer colchicine could suppress this. The non-stem-like cells, they attached less rapidly. The colchicine was also able to suppress their attachment. So this again confirmed that the cells are attaching by a microtentacle mechanism that depends on microtubules. And so in the lower panels here, Monica just took phase contrast images over time, parallel the xCELLigence assays, to show that the non-stem-like cells, even after 3 hours, have not attached very efficiently, where the stems really begin, you start seeing evidence of attachment at 30 minutes. 3 hours, the cells have attached and spread very efficiently. And so this is an important thing to parallel xCELLigence assays with. It's the type of question we get often, to actually see that the cells are attaching and spreading in a way that parallels our xCELLigence assay. And we've seen that very routinely. They parallel each other well. But reviewers will often ask for that confirmation. And the E-view plates from ACEA are a good way of doing this as well. So in addition, Monica actually looked in the mammospheres after 14 days after them, and using live cell confocal microscopy of the spheres themselves, she could see that they formed tentacles on their surface that were suppressible with colchicine, again showing that even after 14 days of sphere formation, those are still forming spheres, and they are supported by microtubules because they're inhibitable by colchicine. Now, the other very interesting thing that Monica did here, very similar experiment to what we did where we looked at cell-cell aggregation. And in the mammospheres, she labeled individual cells with a membrane-targeted GFP via adenovirus. And what she could see was when you label the cells individually, that you can see that they're forming tentacles that extend around adjacent cells and between the adjacent cells in the mammosphere. So while the live cell confocal microscopy yields an image that many people would assume the cells are just sort of via their cell-cell junctions, what Monica's experiment shows is that the cells within mammospheres are actually extending tentacles around each other in a way that can't be appreciated unless you label the cells individually. All right, now there are other consequences to the fact that tentacles are based on microtubules. And one of those being that there's a very common breast cancer drug, Taxol or paclitaxel, which inhibits tumor cell growth by stabilizing microtubules. And so what we've shown is that if you take tumor cells that have very few tentacles and treat them with Taxol, that these cells can develop more tentacles and reattach faster. So this raises the possibility that some of the drugs that we currently use to reduce tumor growth could actually increase the metastasis of circulating tumor cells. And this is something that we'll need to keep an eye on going forward. In just the last year, Rebecca Whipple in the lab done is tried to identify drugs that will target the type of microtentacles glue microtubules more specifically to allow us to target the microtubules that are involved in tentacle formation, but all microtubules like current therapies do. And so what Becky had shown in this paper was the treatment with parfenolide or cosenolide is able to suppress detyrossinated tubulin, but not total alpha tubulin. And the other thing that you can see on these Western blots is that Taxol leads to an increase in detyrossinated tubulin, that in some cell types like the MDA MD157, Taxol treatment leads to an incredibly high level of blue tubulin accumulation. And so this is something to think about going forward. When we look here, what you're looking at is the same field of cells labeled specifically for glue microtubules or total microtubules. Total microtubules in green, glue microtubules in red. And so while all microtubules form this sort of very classic web of cytoplasm, the glue microtubules are a subset of bundled stabilized microtubules that are very clear here at the glue tubulin staining. Parfenolide does a great job of reducing the glue microtubules, but spares the unmodified microtubules so that most of the microtubules in the cell are retained. Whereas treatments like Taxol or colchicine or vinblastine, some of the current microtubules that are available, those target all microtubules indiscriminately and would either destroy or stabilize all of them. Parthenolide and costinolide target specifically the stabilized microtubules. And so again, using xCELLigence technology to test these different therapeutic approaches, what Becky showed here in the breast cancer research paper was that looking here at the vehicle control cells in the open circles, over time those cells attach at a certain rate. If you treat with Taxol, the cells actually attach more efficiently over time. But that treatment with parthenolide here at the bottom suppresses reattachment. Costinolide in the open diamonds also suppresses reattachment, similarly to what we see with broad microtubule inhibition with colchicine in the closed triangles. So this is showing us then the differences between these different therapeutic strategies. That Taxol can enhance attachment while parthenolide and costinolide suppress it. All right, so just to bring this back to sort of the overlying model, epithelial cells form tight cell-cell junctions, and when they're in a confluent sheet, they tend to have weak microtubules expanding outward from the center and strong actin contraction from the cortex. And that this is a situation in which growth is suppressed when microtubules and actin are largely balanced. At a wound edge, microtubules are stabilized into the wound, and actin contraction is weakened as actin rearranges and microtubules are stabilized to help cells move into the wound. So then you get stronger microtubules, weaken actin, and also upregulation of epithelial mesenchymal transition and upregulation of tumor cell growth. This is what we think is happening when cells go into a free-floating state, is that this wound healing effect essentially continues. So you get strong microtubules, weakened actin, and actually increases in stem cell characteristics, because one of the most universal ways to promote stem cell characteristics for many different tissues is to put tumor cells into a free-floating state for reasons that are not entirely understood yet. But basically, in the free-floating cells, the strong microtubules and weak actin promote the formation of microtentacles. Now, something that we've been thinking about a lot more recently is that many of the current cancer therapies that target microtubules, like taxanes and epoxylones, stabilize microtubules and promote strengthening of microtubules. Likewise, inhibition of the actin cytoskeleton is emerging as a strategy to inhibit tumor cell invasion with inhibitors like those that target the ROCK protein and Src tyrosine kinase. But that these actin inhibitors tend to weaken actin contraction, so that both of these therapeutic strategies for reducing tumor growth and local tumor invasion lead to the same types of cytoskeletal tertubules and weak actin that we would expect at wound edges, and then also potentially increase stem cell characteristics and increase metastatic potential. So this is something that we really need to think about going forward in terms of, are some of the strategies that we're using to inhibit tumor growth potentially making metastasis worse? And there is some emerging evidence for this. There's publication in 2012 from a German group, Katharina Pachmann's group, where they basically looked at patients who were given chemotherapy before surgery, so neoadjuvant chemotherapy. And what they did was they followed very carefully whether circulating tumor cells decreased in those patients when they were given therapy, or whether circulating tumor cells increased when the patients were given therapy. And so what they found was when patients were given chemotherapy and their circulating tumor cells decreased here at the green line, that after 7 years, 100% of those patients were relapse-free. But that if circulating tumor cells increased in patients when they were given neoadjuvant chemotherapy, that after 7 years, 95% of those patients had suffered relapse. So this is suggesting that we need to look very carefully at what the effects are of these therapies on circulating tumor cells. Because if we just think back to the very first slide that I showed, since our threshold of detection for tumors in patients is a foci of 10 million tumor cells, when you give those patients therapy and the tumor shrinks on the MRI, is the tumor shrinking because the tumor's dying, or is the tumor shrinking because it's scattering? And can our current detection methods tell the difference? They really can't. And so what this study here is showing is that when CTCs increase during therapy, which would be an example of a scattering model, that those patients may be at much higher risk of relapse. And so this is something to think about going forward. All right, so then just in closing, I just wanted to show you some of some more exciting unpublished data that we've had over the last year, where what we've been doing now is taking surgical samples from breast cancer patients immediately at the time of surgery, placing them onto the confocal microscope and imaging their membrane dynamics right away. And so what we can see is that we can detect cells with microtentacles here within hours of surgery on the patients. And so looking here at low magnification, we can see several cells that are forming tentacles. If we look at higher magnification, we can see that these tentacles are very long and very similar to the morphology of tentacles that we've seen on cultured cells. But even more importantly, here from the fresh cells from patients, we can see tentacles on those cells, but then in the same cell, when we treat with a microtubule depolymerizer, we can see suppression of the tentacles in those cells. So this then gives us the ability to test drug responses in cells that have been freshly derived from individual patients that will eventually support a goal of personalized medicine in that we can test drugs specifically using the cells from individual patients. And so finally, just working with our engineering collaborators, we've developed different ways of quantitating these tentacles in a very rigorous manner so that we can classify tentacles on the basis of their length. Here, those that are longer than 4 microns have a red dot at their terminus versus those that are shorter than 4 microns have a green dot. And this allows us to get metrics for microtentacles like average microtentacles per cell, average microtentacle length, that are the kind of thing that I've never been able to convince graduate students or postdocs to measure on the microscope by hand. So now at least we have a computerized method for making these measurements. All right. And so then just in closing, I'd just like to thank the members of my lab who performed all of these studies, our collaborators at other institutions and funding agencies that supported this work, but also the ACEA xCELLigence technology has really been instrumental in helping us quantitate the efficiency of tumor cell reattachment, both with the EMT genetic models, looking at the stem cells, and then currently as we're developing therapies to inhibit tumor cell reattachment. And then Monica Charpentier in the lab was also supported by an ACEA travel award for presentation of her stem cell work at the Keystone meeting this year. So thank you very much. I'd be happy to take questions at this point. I guess